Today I'll be discussing APOE and its role in the development of Alzheimer's disease. Our learning objectives are to explain the basic role of APOE in normal CNS function. We'll identify some mechanisms responsible for the role of APOE4 in development of Alzheimer's, and we'll examine the differences in Alzheimer's presentation within patients who carry APOE4 versus patients who don't. For an overview of Alzheimer's, it's a neurodegenerative disease primarily characterized by a progressive loss of memory and other cognitive functions. The hallmark of this disease includes a buildup of amyloid plaques and neurofibrillary tangles, which will eventually lead to the death of neurons. The severe degeneration of the brain that we see in this disease typically begins in the mediotemporal lobe, which includes the hippocampus and entorhinal cortex. As the disease progresses, we see it spread to other structures such as the neocortex, which includes the temporal and parietal lobes, the frontal lobe, and in later stages, the occipital lobe. APOE is the gene responsible for the creation of apolipoprotein E, which is the most abundant apolipoprotein in the brain. Its primary role is to transport cholesterol and other lipids to neurons, where those lipids play a role in synaptic function and injury repair. In the brain, APOE is mainly produced by astrocytes, but we do see it expressed by other glial cells, such as by microglia. Over here is the general structure of a lipoprotein. We see the phospholipid layer, which encases the hydrophobic cholesterol and triglycerides. Over here, we see the apolipoprotein, which wraps around the phospholipid layer, giving it structure and allowing it to move within the aqueous environment of the brain and body. The predominant APOE alleles in humans are APOE2, APOE3, and APOE4, each having a different impact on the risk of developing Alzheimer's. APOE2 appears to reduce that risk, APOE3 carries a neutral risk, and APOE4 is a major genetic risk factor for developing Alzheimer's. The frequency of these alleles will vary by ethnicity, but in general, APOE3 is the most common, followed by APOE4, and then APOE2, which is the rarest allele. Each allele codes for a slightly different isomer of APOE, which results in the different impacts on Alzheimer's disease risk between each isomer. APOE is a 299 amino acid sequence. The differences between the three isomers are in only two positions on that whole sequence, position 112 and position 158. APOE4 is implicated in several key areas of Alzheimer's pathology, which include the development of beta amyloid plaques and neurofibrillary tangles. We also see its promotion of neuroinflammation, dysregulation of lipid metabolism, and a breakdown of the blood-brain barrier, but we'll only be focused on the plaques and tangles for today. APOE4 promotes plaque formation in several ways, including by accelerating early seeding of beta amyloid. So in Alzheimer's patients with APOE4, we do see the presence of beta amyloid plaques in earlier stages versus patients who do not have APOE4. It also enhances fibrillogenesis, which is the aggregation of beta amyloid fragments into beta amyloid plaques. And we also see a reduction in the ability to clear beta amyloid once it begins to build up in the brain. APOE4 also enhances deposition and negative effects of neurofibrillary tangles by increasing tau phosphorylation, which is essentially the increase in the amount of tangles within the neuron. It also exacerbates tau-mediated neurodegeneration, which are the negative effects that the tangles have on the neurons that eventually lead to cell death. So APOE4 as a whole does increase the risk for developing Alzheimer's. It is currently unclear if APOE4 causes more rapid cognitive decline, but the general consensus is that Alzheimer's patients with or without APOE4 tend to have a similar rate of cognitive decline. Where the differences begin is in the presentation of that decline. Carriers will typically suffer greater memory deficits than non-carriers, while non-carriers will suffer greater non-memory deficits, such as in executive functioning. These deficits seem to correlate to the differences in volume loss in different areas of the brain. In patients with APOE4, we see greater degeneration of the mediotemporal lobe, which will include the hippocampus and entorhinal cortex, which correlates with the greater memory deficits that this group experiences. In non-carriers, we see greater atrophy in the frontoparietal lobes. So degeneration in the frontal lobe would correlate with the greater deficits in executive function that this group experiences. So in general, these effects seem to demonstrate the variety of complex mechanisms involved in Alzheimer's and really elucidate the need for research beyond our current hypotheses. And hopefully in the future, we'll have more research on lipid metabolism and its implications within Alzheimer's disease. And these are my references. Thank you.